another area of research in my lab has been to develop new DNA sequencing technologies. And in fact, we were the first to show how to sequence single molecules of DNA. That led to the founding of another company called Helicos Biosciences. And in fact, their product, which is shown here, was the first instrument that by itself was capable of sequencing an entire genome. And I, in fact, sequenced my genome on it, or rather Norma Neff did. And so one person, one machine was able to do an entire human genome. And another student, Dmitry Pushkarov, did the informatic analysis. And that, I think, is a real game changer for the field because it democratizes the technology. And it means that anybody can get in this game and unleash their creativity on the problems that interest them. And I think it's going to be good for science. And it's not stretching the analogy too much to say that the case in biology now is that you can have your, you can go down to the store and buy your particle accelerator, and every lab can have one. You could have several at each university. And that's sort of been recognized, this tremendous innovation that's happening in the field of DNA sequencing by a number of companies. And this was a figure from Nature magazine a few months ago. They had their 10th anniversary of the Human Genome issue, and they're showing the cost of sequencing decreasing very rapidly over time, and the number of individuals going up. I happen to be in the first handful of people to see their own genome. That's me up there. The technology is decreasing is sort of decreasing in cost in a really phenomenal way. And this is it is staggering. Okay, this is a chart I made up when we published our paper. And you can see by whatever metric you care to look at, whether it's the cost of the reagents, or the number of machine runs, which is a measure of the capital cost involved, or the number of authors on the paper, which is a measure of the manpower costs. Everything is improving exponentially. And none of this has stayed static. So this was 2009. Since then, and as I mentioned, this was sort of, in our work, it was sort of marked this transition to the point where you could do it with a single machine, and you didn't need a genome center to do it. And so now it's become the industry standard to do that. And all of the competitors, Illumina, ABI, are introducing machines that will allow you to do the same thing. And so the sort of bitter commercial competition has been a great spur for the scientists involved in this area. And it turns out that all these machines have different properties. They have different strengths. We use, in my lab, machines from virtually every manufacturer for different things. And so the last thought I'd like to leave you with before we sit down for the discussion is that these DNA sequencers are useful for much more than just looking at human genomes. And as they're getting out there in people's hands, you're seeing all kinds of applications. And just in my lab alone, here's a few examples. We got interested in using these machines for things like non-invasive diagnostics. We demonstrated the first non-invasive diagnostic for Down syndrome, prenatal diagnostic for Down syndrome, to make amniocentesis obsolete. And that's now something that has been sort of taken up by a number of groups and companies working in the area. We've been using similar approaches with sequencing to predict rejection of organ transplants. We've looked at things like the immune system, okay? Your genome you'd sequence once in your life, and it's not going to change that much, but your immune system is always interacting with the environment and changing itself in response. And you can, in principle, sequence that over and over again every month if you like. And if you're smart enough, perhaps use that sequence data to say something about what you've been exposed to and what your body is doing. And so we sort of initiated that field by sequencing the full antibody repertoire of a model organism, zebrafish, since going on to look at humans. And this is something, again, in particular, as new sequencing technologies come out, we're going to adopt them. We're awaiting delivery of a new machine from Pacific Biosciences, which is sort of the second generation single molecule sequencer to come on the market. And it has properties that'll make it very good for that sort of measurement. In 2 weeks, we should be getting it, so I'm really looking forward to that. And then, of course, I've spoken a bit about my genome, and I'll just say, sort of maybe a measure of the lack of creativity in the field, that I wasn't the first person to sequence their own genome or have it sequenced, but I was the first one to go ask a doctor what it meant. And that led to this paper on the clinical annotation of the human genome. And I was sort of the index study. And a couple dozen doctors at the Stanford hospital went over my genome and tried to give me advice. They wanted to answer this problem. How do you treat a patient who walks into their office with a genome in hand? And we took a shot at that. And there's very interesting stories about that. And we can get into it in the discussion if you like. And so I'll stop here, and we can maybe get the other panelists up.
Thanks, Steve. So, remarkable technology, and the question is, how do we translate that into improving healthcare? And so to address some of those questions, I'd like to introduce the panel. The panel. Great. Sue Desmond-Hellmann and Brook Byers. Welcome. Thanks. So Sue joins us. Sue is Chancellor of University of California, San Francisco. She is a really interesting person. That her prior life, she was president of Genentech Development, involved in the discovery and application and marketing of a number of drugs, including Herceptin, Rituxan, and other of these marvelous biotechnology drugs. Next is Brook Byers. Brook is a venture capitalist, and Brook is one of the co-founders of Kleiner, Perkins, Caufield, and Byers. He's been involved in over 50 biotech companies, their founding, including Pacific Biosciences that was mentioned earlier. So I'll leave the panel to discuss, and great. Thanks, guys.
Go for it.
Yeah, thank you, Rodney, and thanks for inviting us all to be here today. So Steve, Sue, and I were talking about how we should approach these topics. There's so much to talk about, and so the first thing we've asked is, can we get 3 hours to cover it all? Because first let me kind of define who we are and what we do. We're in the life sciences, but we're driving the whole interdisciplinary and applying all the different tools, just as Steve elucidated in some of the work he's doing. So the three of us take a systems view toward medicine and biological sciences. And so in talking to many of you who I know are working on digital problems or climate or environmental problems or engineering problems, to put it in those terms, we think of our bodies as biological networks, that disease is when there's a dysfunction in some of those networks. And we work with the window to that, which are blood and saliva, which give us a look into things that we can measure and even predict. And so this gets us to disease stratification, progression, and tightly managed treatment. Tightly managed treatment is the key to good outcomes. So things we can talk about today, and we're going to go really fast through them. And then when we get to Q&A, you can be thinking, if you want to get into some of these others, are new high-value molecular diagnostics, drug therapeutics that are targeted, stem cells, sequencing, global infectious diseases, healthcare delivery systems and healthcare IT and how that all ties together, healthcare economics, and public policy. I thought I'd start by going back to sequencing, since Steve was on that. And to put it in digital terms, what you saw, Steve, on that chart that Steve put up is that the ability to sequence and the cost of doing so is moving faster than Moore's law. And in the first generation of sequencers, they were really good at read length and runtime, but not good at throughput or cost per base. The second generation of sequencers were good at throughput, 25 gigabits per day, and cost per base, but not good at read length or runtime. Read length is the length of DNA sequence that can be read at any one time. And so they had to be spliced together using bioinformatics. And so in came computation and all that need. But by splicing it, they didn't literally put them together, as they overlapped them digitally and with IT, but that introduced error. And so the third generation, which are the ones that Steve's working on, and he's going to get a good one next week, it has gone back now and improved on read length, with longer read lengths, less error, and less chance of missing polymorphisms, which is where you find rare events, which is going to be important to disease. Runtime, so speed, and cost per base, and throughput. And so for those of you who are computationally oriented, they're going to be 250 gigabits per day coming off these single molecule systems. alert there. Huge computational problem that's not been solved that will need to be done in the cloud, and how all that's linked and how that data is held and managed and stored and done all that. That's all part of the future that we can get to. So Steve, if we go back to you, you commented on that you took your sequence to a set of doctors. Was the value in the pharmacogenomics or what else?
Yeah, so I think the two most valuable things to come out of it were the pharmacogenomic analysis, where I was given a list of drugs, which if a doctor were to try to prescribe them to me, I should say, well, maybe not that one, or if that one, different dose. And there is pretty good literature on the drugs, and the interpretations are reasonably straightforward for individuals because they have to do with either variation in the target of the drug or in the metabolism of the drug, and so straightforward biochemistry. The other useful piece was the really rare variants, and so it's been estimated that each of us carries half a dozen to a dozen very rare variants or mutations with us that link to various diseases, and it would never be economical to screen anybody for them. But when you have the whole genome, whichever one it happens to be just pops up. And in my case, it was related to cardiomyopathies, and so led to a very interesting dialogue with our cardiology department at Stanford, and something that I think the economic benefit will be that you'll be able to test people at risk more frequently and people not at risk less frequently, and thereby save costs and save people the trouble of unnecessary tests.
Now, so let's go into dispelling some of the myths and some of the confusion that comes out about this area. What is the value of sequencing one person, and will there be a need to sequence 10,000 like the project in England? To get value?
It's a bit controversial right now. This idea of common variants, common disease, and it's taken a lot of heat in the press. Nicholas Wade had an article in the New York Times saying that, the Human Genome Project has failed to deliver any medical benefits 10 years on because it's been so slow working that aspect of it out. But I think that's a too narrow a view of what most of us would have said the benefit of the Human Genome Project is going to be. I think most of us would have said, we don't know what it's going to be good for, but it's going to enable a lot of new things. And one example would be this prenatal diagnostic thing we developed in my group, because we couldn't have done that without the reference genome, and it had nothing to do with finding mutations or anything like that. It just had to do with the ability to have that sequence out there and map short snippets back to chromosomes of origin. So that was something nobody would have predicted. It was a very tangible medical benefit to derive from it.
Agreed.